Great. Well, thank you all for coming here today to the VICC Virtual Molecular Tumor Board monthly call. We have the honor to have Dr. Alper Uzun here today to present to us about a tool that we can use for variant visualization and analysis called Variant Graft Craft. Dr. Uzun is the Director of Cancer Bioinformatics at Legarreta Cancer Center at Brown University. So please take it away, and thank you so much for sharing this with us today.
Yep. Thank you so much, Beth. It is really honored to have my presentation here. So let me share my screen very quickly. So can you see my screen right now?
We can see your screen. It's the PowerPoint.
Right.
Yep.
Now I'm gonna be putting on presentation mode. So it's still visible, right? Okay.
Yes.
So yeah, again, thank you so much for having me in this meeting. So today I'm gonna talk about one of our recent tools. It's about Variant Graph Craft, which is the name is Variant Graph Craft, and it's about VCF file visualization and analysis tool. So before getting into this, I would like to introduce our lab, what we do and who we are. So we are a computational biology and bioinformatics lab, and we focused on understanding the genetic basis of complex diseases. So when we call about complex diseases, we are particularly working on cancer, but including Parkinson's, preterm birth, preeclampsia. We already studied those diseases, but currently I can say 95% we are working on cancer right now. And what we do is we study this genetic architecture of these complex diseases, as I mentioned, especially cancer. And we are using network biology to understand this genetic architecture. So we are mapping those gene-gene, protein-protein interactions and their disease relationships. And we are, again, developing tools for visualizing the genetic variation, biological networks, and also we are developing tools like user-friendly, especially this is our motto, like user-friendly tools for single-cell sequencing analysis. And we are applying machine learning to uncover these hidden patterns. So in these spheres, I just want to pinpoint what we really do in those fields of bioinformatics and also genomics. And of course, like nobody works alone. So this is our lab members. So we have a bunch of different uh, backgrounds, like from bioengineering to medical school students, from from fellows to residents, computational biology, computer science. So we have bunch of different backgrounds, and this is very helpful for us because then we can move in several directions with combining our background information for that kind of purposes like genomics and bioinformatics. And now if I talk about like the VCF files, so most of us know, but this is kind of general intro that I would like to use. So when we talk about because nobody has to know what VCF really for any perspective, but variant code format is a file format for genetic variation data. And this scary part of this, the black screen, maybe not for the coders, but most of the clinicians or wet lab scientists, they don't want to deal with all this kind of representation. And in this representation, this is very highly structured text file. It is packed with essential genomic information such as variant positions, alleles, genotype codes, quality scores, and everything in this matrix. And if I ask you, okay, bring me this gene and these related variants, there is no way for a kind of regular person, which means like who doesn't have any kind of coding background, dive into this, text files and getting. So there are, of course, so many tools like that. So the question is why we needed a better variant visualization tools. They're already existing in the field of, genomics, bioinformatics. So this is really coming from our own needs, literally. So for our own genomic research, including whole exome sequencing, targeted panels, whole genome sequencing, we realized that there are tools, of course, like available, and there are very, very powerful, good tools for VCF analysis and visualization. But they are fall short in several ways. For example, they have fragmented workflows. What I mean is there's no single tool covers all essential functions like annotation, filtering, visualization, but users or like in the lab, the bioinformaticians, they need to combine these multiple tools. Or if you don't have a bioinformatician or a programming person, then you are depending on whatever existing with your own needs. So there's a steep learning curve. So many tools are not designed with usability in mind, require command line expertise or complex setup. So most of the time, even the very first step, installation is very tough. You need to put these dependencies, no, do these dependencies, that dependencies. While you are doing this, you are losing time and you are losing the interest of what you are doing also. So, and also this is exactly direct observation from my lab as well. Although we are bioinformatics a lab, we have we had research assistants who were, for example, visiting hospital, enrolling patients, getting their blood samples. So these research assistants specifically working for that purpose. And again, they don't have coding background. So that's why from my direct observation, we need kind of more user-friendly tools. And then the other thing is limited accessibility. Again, not everyone on the team, especially those without computational backgrounds, can use them efficiently. Like exactly the same example that I gave, like they are going and enrolling patients that they don't have to know all this coding. And then workflow inefficiencies. There are times spent stitching together tools take away from actual discovery. You need to know, okay, after this tool, I'm going to use this and then this. So this cartoon on the side, it shows like what if in the cell lines, what happens. So this is kind of a view of what's going on from the cell lines perspective. So again, these VCF files with their extensive data richness are one of our essential resources, but their complexity presents a notable challenge. So I just want to give a little bit history about how we came to this VGC point. So this visualization and interpretation of this data demands both resource and expertise. So here, what you see on the right side of the screen, filtering, extraction, phenotype annotation, plotting, all these steps needs different tools, this different expertise. So what we did was, what we did previously before VGC, we developed another tool. It was called Viva. So you see the paper here. It's a visualization of variance. Again, another variant tool for visualization. We built this and published in 2019. So this was not, unfortunately, like user-friendly that we were planning, because this was, again, command line. Only thing that we did was probably Jupyter Notebook. It was at least easing the pain. So you were using the Jupyter Notebook as a user. But then the interesting thing here, so before publishing this, we put this on the bioarchives, literally in 24 hours. This was one of the top 5% bioarchives, like preprints or tools just showed up in less than 24 hours. And we didn't know that it was gonna getting that kind of attention because there are already existing so many tools. One of the interesting thing was we used Julia programming language. Julia programming language, one of the fastest programming language after C++. So Viva was the first tool for Julia programming language for bioinformatics or biological purposes. So this was under the attention of Nature, and they did an interview with us because they were interested in Julia and how Julia was fast and how it can be used in that kind of, life sciences. But this was a good adventure, I would say, because after that, Julia programming language is fast, but on the other hand, there's no that huge community. And the other thing is, whenever you have a new, for example, version, all the dependencies are crashing. You need to update everything, or you can't do any kind of with the existing version for the previous version. So basically, it was kind of challenging after you publish everything. So this version available on GitHub and it is working. But if you want to add more features, you need to update everything. So we thought, okay, we are going to be not using Julia programming language anymore, but we'll be moving forward, maybe Python, maybe Java. So, but also we need to make a better, nicer visualization tool. So we came up with variant graph craft. You can think about like what kind of name this is. We are crafting the graphs, but the craft part is probably coming from Minecraft. Our 11-year-old son, when he was playing at this publication, like they say 10 years old, he was playing Minecraft, and I was saying, "Oh, craft. Yeah, we are crafting graphs. Yeah, let's make the name. You will see why this is going to be like this, Minecraft." So you see these three people, Dr. Carniero, Dr. Ece Uzun, Dr. Lauren Messingham. So they're all working on the clinical side of the field. So Dr. Carniero is a medical oncologist, and he doesn't know any coding or any kind of bioinformatics related field, but he's very, very good medical oncologist. And Dr. Ece Uzun, clinical bioinformatician, she's very, very good at coding and also very good at in the clinical side. And Dr. Messingham, she's clinical geneticist, but she doesn't have coding skill set, and she doesn't have to also. But they were very, very useful for letting us know how should we move forward for a user-friendly tool. And Jennifer Lee and Andy Wang, these are wonderful students. Jennifer Lee, she's the leading programmer, and Andy, he was focusing on integration of evidence-based data. So with the help of Dr. Carniero, Dr. Uzun, and Dr. Messingham, we developed this tool by their feedback. For example, as a clinical geneticist, Dr. Messingham always telling me that she has a trouble in telling the story to the patient when she was talking about a specific gene location or a chromosome location, because she was showing a kind of default gene or chromosome in order to tell the story. So she gave her feedback for us, and Dr. Carniero as well. And Dr. Uzun, not only for clinical side, also in the hardcore, like how we can apply some coding parts of that. So basically, these three people were very efficient for us to move forward. And they are my colleagues. So not only friends, but also colleagues. So this is the landing page of variant GraphCraft. So what you see is the VGC QR code, or you can simply go to this link as well. So what we are doing is, so we try to make a kind of explicit GitHub page, how you can move forward. So basically you are downloading the tool, then the only requirement is 1.8 or higher Java version. So everything is based on JavaScript on the front end, Java backend. And if you have operation system for Apple, Windows, or Linux, all three of them going to be working fine with variant GraphCraft. And when you download the program, so this icon, VGC app, is only button that you need to install the machine. So you don't need to open the command line, writing the install.mod or any kind of thing. You just click and installing. And if you have 1.8, it will work. If you don't, then you need to update your Java for the required version. So this is the landing page, as I mentioned. What you see is here, potential options and uploading your VCF file if you have a build 37 or build 38, and some introduction and documentation backend for the use of VGC. So the workflow, I'm going to tell you like as we move through, but this is the foundation of the tool. So if you have a gene as a search query as a gene or genomic location, or if you have multiple genes or multiple genomic locations. So if you have a gene or genomic location, you can simply enter your information, or if you have a file including the gene names or genomic locations, you can upload this as well. So what's happening is, first, there's a retrieval, like extracting this information and building the info and index files. And then variant extraction starts. If you have a huge file, like 20 gigabyte, 30 gigabyte of file, it may take some time, like a minute or so, in the very initial opening, like initial uploading the file. But after the initial file uploading. If you are working with this file for the second time, third time, the next times maybe, let's say, then it will build the file like and ready to use in seconds. Only for large file, initial is taking some time. So what you do is you can visualize the variance as a bar graph or node graph. I'm going to tell in detail what they are. Or you can download the data in Excel file as a variant data. Or you can create genotype testing for heat map for deep depth of coverage. And you can compare samples. So based on your samples, case or controls, or group 1 versus group 2, whatever your groups are. And then you can annotate these specific gene or genes or genomic locations. And this is the integrated database and packages. So we are using this knowledge base support information from MSIC database, CleanVar, Nomad, and for the human genome assembly, we have 37 and 38 builds. And the libraries and packages supported by recharts, SynFusion, sorry, and React for force graph we are using for the node graph visualizations. So we also thought about safety and compatibility. For example, you have this VCF file. When you are uploading a cloud in order to do your analysis, basically cloud is somebody's computer and you don't want to upload your valuable information, genetic information or genomic information on the cloud. Even if it's de-identified, you may not want to. So that's why there's no cloud uploads. You are installing this tool on your machine or on your servers. And this is ensuring data privacy in your own environment. And users can work with their genomic data in a secure and confidential setting for that reason. And we are supporting, as I mentioned, human assembly HG37 and HG38. And here, what you can do is, as a user, you can enter a gene name or a specific genomic location and to search for those specific genomic locations or genes. And then also VGC supports visualization of pathogenic variants. So basically, this pathogenic variant idea is coming from Dr. Uzun. So when we were discussing in our meetings, she was saying that, okay, we are showing all the variants, all the pest variants, like validated variants, but what about the pathogenic variants? We need to include them. So this was a wonderful idea, and we include this feature. So the data is coming from CleanVar. So when you visualize the, I'm going to show you, but when you visualize the data sets, you can specifically ask for pathogenic variants and it will show up. And also VGC enables the incorporation of phenotype input data to visualize genotype-phenotype relations. And the graph craft is coming here, where I was just mentioning the crafting the graphs. So you can visualize all these variants and patient or sample relationships in a kind of graph visualization. So in order to in order to show how this tool works, I'm going to using from one of our studies. We also used in the paper as well from preeclamptic patients and term mothers. So this is demonstrating the VGC capabilities. This is about preeclampsia, and preeclampsia is a hypertensive disorder of pregnancy. It's associated with high risk of hypertension, cardiovascular disease later in life. So when the pregnancy starts around 25th week, or this is early onset, or late onset around 34 weeks, blood pressure of the mom is increasing really high, and the liver enzymes showing really high values, ALT, AST. And the only solution is the baby should be delivered. So around 24-25 weeks, unfortunately. And this is called preterm birth, or 34, still preterm birth, early or late onset. So we work with those mothers. And my research assistants, they were visiting the hospital. We have Women and Infants Hospital, one of the affiliates of Brown University. So they were enrolling those mothers, preterm mothers and preeclamptic mothers and term mothers. And we collected 143 samples, 61 of them early onset severe preeclamptic cases, and 82 of them are term mothers which is like our controls. So what you see is here in that small window, this is 2.45 gigabyte of data. So there are 143 patients in this VCF file. And after quality controls, after filtering everything, now there are 700,000 variants existing. So in these two screens, these are each bars showing up each chromosome location. And on the right side, red color show the pathogenic variants. So you can see like in chromosome 1, we have around 50,000 variants. And as a pathogenic variant, there are 40 some, like around 40 variants for chromosome 1. So this is a kind of not live demo, but like the feeling of live demo. So let's say you are entering this BRCA1 gene and you are asking the question, okay, bring me all the BRCA1 gene related variants. Then it will bring up all the variants in the BRCA1. So the number of, you see the number of variations and the height of these bars showing like the variants in that specific location. And if you go in deeper and deeper, when you click, you will see different colors. If you see the darker blue, this will be a variance in the exonic regions. If it is lighter blue, it will be variance in the intronic regions. So you may ask this whole exome sequencing, how come that we will be having intronic regions? So still we have some exome intron boundaries, and there are some variants captured in those regions. So that's why we have intronic regions. So if you go deeper, by the way, everything is user friendly and interactive. You can click and go in more detail for each bar here. So when you click here, there will be an information like showing up, okay, this is a benign variant and this is where it is located. And these are the potential conditions for that specific variant. And then this is the whole view. If you click more in deeper, you will see on the side nomad visualization, nomad link, and immediately it will be showing up the variant associated with this nomad data, and nothing is transferring to nomad, only the location of the variant. So if you want to move from here to nomad, you can click any of the features here, and then you can move with the nomad. Then the next thing is, what if the variants in this gene BRCA1, you just want to extract to your data as an Excel file. So the variant data here, it is showing in that bar. So it is going to be showing up as an Excel file. And you can save this as an XLX, XLS, CSV, and PDF. As if you are in the Excel file, you can create your existing like additional sheets in order to, for example, add more data here, whatever you like to do. But everything is whatever in the VCF file extracted and nicely visualized or ready to move forward for example, post-analysis, whatever you are interested in, you can carry this file. Then the fun part starts, like the node graph. Okay, now we have this BRCA gene, and you, for example, you already submit a file. It's a phenotype file. It is very easy. You are just describing what are your cases, what are your controls with the IDs. For example, here, sample group 1, group 0, and the sample group 1. Immediately, you can visualize your patients and the variants. So the blue dots are the variants. The human figures, red and green colored, or you can choose any other color. It doesn't have to be red and green. And these are showing the different group phenotypes. Let's say cases and controls. And then if you click on a variant, you will immediately see if this variant is benign or pathogenic. And also on the left side, you will immediately see the nomad information. Or if you click on a patient, you will also, sorry, this is the patient and this is the variant. I need to see better. So if you click on a patient, you will see the patient ID. If you click on a variant, you will see immediately variant information. And this is like 2D. But if you want to create a 3D, you can simply click on the 3D and you can start to visualize this like a kind of in the structure biology, working on a protein structure, you can visualize all these variants and associated patients with this specific, 3D environment. Moreover, here you can only visualize homozygous reference, heterozygous variant, or homozygous alternative variant. These are all possible here. For example, here, let's say you are only looking for pathogenic variants, like this is from Dr. Uzun's suggestions. So in the pathogenic variant, immediately from here to, you are going down to here because these are all pathogenic variants, and you can easily see what kind of, for example, groups like Ks and controls shared these pathogenic variants. And again, you can play around. There are so many other details, but I'm not going to take the detail, but you can play around only heterozygous, only homozygous alternative, or only like highlighting the edges of these interactions. You can visualize that too. Then the next thing is compare samples. For example, in the BRCA one, we have all these 52, 57 variants. What if we apply genotype testing, and what if we want to visualize the depth of coverage for those variants? So lighter blue, like in the ocean, on the surface, it is lighter, and if you go deeper, it is darker. So darker means more depth of coverage, lighter, less depth of coverage. And you can see the variant information here, and their adjusted p-value and then nominal p-value. If you click any of the arrows here, you can visualize the number of genotype information and comparative analysis, if it is significant or not, for the homozygous, heterozygous, and homozygous alternative. Then the next question is, what if I have a specific genomic location? I am not interested in a gene, but a specific location. The format is very common for whatever you are using in other tools, like the chromosome number, starting and ending position of this variant. This is a specific one variant. Or if you grab a bigger, genomic location. So it's up to you. It's up to your interest. Then you will see the specific variant and then their visualization in a kind of graph environment. And you can play around from this step on. If you want to upload several genomic locations, you can upload a file. Or like in the BRCA1 gene, no, you are not interested in one gene. You are interested in 10 genes, 20 genes. Then upload a file and see them in the parallel view. So about this genomic location, like I was telling you, what if you are only interested in heterozygous ones? For example, this is everything, as you see. And what if you are interested in only heterozygous variants? It will show up. Then, or homozygous alternative. So this is a kind of option that you just want to move forward. And you can save all these images for your publications or for your presentations. Then the next question is, okay, now you know the gene, you know the variants. What if you want to learn more information about this specific gene? For example, if you upload a file about genes, like the way you see the tab, it will be several tabs for each gene. So for example, in BRCA1, in our example, you will see all the pathogenic variants, benign and no consensus variants as well. And also you will immediately see the data about like the Go terms, like what is the biological process of this gene, or molecular function, or cellular component, or any pathway associated information existing. So this screen capture, basically this page is scrolling down. You will see more information here. So at the end of the day, I can tell you that this is a tool that it's a kind of comprehensive solution. It's a kind of complete set of different tools that researchers may want to use for their own analysis. And we try to make it as much as possible user-friendly interface. So it's enabling a easy navigation and operation, and from different perspectives, from different background of people. And you can create custom queries, like the way I was describing, user defined groups, different gene groups, different genomic positions. And it is a kind of integrated variant querying, which means that you can include features of querying variants from different public databases that we were using, MSIG database, Nomad, 37 and 38 builds. And we are trying to make enhanced visualization tools like supporting by histograms, spreadsheets, node graphs, and heat maps, and almost, I should say 95% everything is interactive. It's not static. So again, the VGC is available from GitHub, and you can use this QR control and QR code as well. The other thing is, again, nobody works alone. So this is my group, Genomic and Information Intelligence Lab members, and these are my collaborators. So we are, we are a kind of lab that we are looking from every perspective, from fellows perspective, from undergrads perspective, or from very, well equipped lab PI's perspective. So any kind of. And I would like to thank you. And if you have any questions, I'm very happy to answer. And by the way, those cartoons that you see, these are coming from me. Actually, besides being a scientist, I'm also a professional cartoonist. So every cartoon like here, biocomicals.com is available. If you want to present in your, talks, presentations, they are all free. So also you can follow the new comic cartoons from any topic about medicine and biology. So yeah, this is my end of my talk. So I'm open to questions. Thanks so much.
Thank you so much. I'm intrigued about cartoons now. This is great. But also, it kind of fits right into the theme of visualization and being able to kind of teach what we're looking at from a visual perspective. I guess I see a lot of utility in this. Like if a lab were able to take all of the pathogenic variants that they had seen in a specific gene, it might be a really great tool for discovery of important domains, things like that, to or just a way to create resources for laboratories to use. So thank you. This was really great and gave a lot of really nice insight.
Thank you. But I think I forgot to mention one thing. So some VCF files, carrying one sample. Of course, you can visualize one sample as well. But the ones that I gave these examples, in one VCF file, we have these 143 samples. Also, we visualize 900 samples. But the thing is, like, it depends on your needs. If you want to visualize a multi-sample VCF file, this is specific for that purpose. It is very efficiently working because you want to see those Kaizen controls or different groups. Or one sample is going to be still going to be working efficiently.
Cool. Thank you. Are there any other questions from the audience or comments?
If I may ask something, thanks a lot for the talk, Dr. Uzun. Are there any examples that you can show with the use of the heat map? I guess one way would be to download the file and then use something like ggplot to produce a heat map.
We didn't use the ggplot, but this heat map is, for example, in your VCF file, whatever the variance and the depth of coverage in the file, it is immediately taking this information and turning into a kind of like the color grading based on the depth of coverage, but also especially if you have again multi-sample VCF file, you can apply genotype testing for a specific variant, like basically abundance of, for example, homozygous in cases versus controls. So you can visualize this information as well.
Right. Thanks.
Yeah, you're welcome. Thank
you. Any other questions? Otherwise, I have one. Have you heard of other laboratories or groups using this tool with success, and what kind of projects have come from this tool?
Well, this is a kind of new tool, but we started to receive emails, especially some core labs, for example, they are asking about some features and existing features, but also they are suggesting some new features, or sometimes they have trouble installing, and the problem is Java. For example, they don't aware that they have 1.8 or higher, and as soon as we tell them it is feasible. And also from like all over the world, I think mostly core labs for research purposes, people are approaching us to use, and mostly bioinformaticians. I didn't receive any email from a non-com bio person.
Mm-hmm.
Yeah.
Great. And then the last question I have is, do you plan to keep this tool and future iterations as publicly available?
Yeah. So basically, this current version, the one that I showed you, this was released in, I think, late September or October, something like. So this is the latest version. And in our next versions, we want to implement like machine learning in order to prioritize variants and some additional better visualizations, especially for phenotype genotype relationships. And the other thing is, the question is, although okay, the purpose was like making a secure, safe, tool. You don't want to upload your data. Also, what if we make a kind of, again, secure web server, but de-identified data, and you will be entering with your, for example, a specific registering system. What if we made it like on the cloud? So
Mm-hmm.
this will be a little bit away from security, because now you are uploading your data. This is still we are thinking, but we have some preparations for applying machine learning in order to prioritize. This is one of the main
Mm-hmm.
ap- application of the version.
Cool.
Yeah.
Yeah.
But also we are very open to suggestions, because the thing is, for example, as a computational biology lab, and also actively working from the, like doing the sequencing analysis, collecting these patient samples, everything, but still we need some outsider user information. For example, like Dr. Uzun was suggesting these pathogenic variants, or like Dr. Messingham, she was asking, what if we make it like more visual in order to tell the story to the patients? So there are still open kind of topics that we need to make it better. So especially in this community, if you have any suggestions, I wish you had this, or we need to have that kind of application. I'm very open to, and very happy to discuss more detail for some
mm-hmm.
potential features.
Great. Yeah, the first thing that comes to my mind is kind of the, integration of current guidelines, like our classification guidelines. But I know that those are pretty nuanced and they're not perfect, and you'd probably need additional information from outside. But I know that some other like commercial groups bring in like a automated classification, and none of them are perfect, but it's a nice aid when you're looking at a variant from an end user perspective, I think. Oh, great. I see we have a question. Go
Yeah,
ahead.
can you hear me?
Yes.
Hi, that was a really nice talk. Thank you. I just wanted to ask if you have tried VGC with visualization of indels and CNV, especially for cancer, because it gets very complicated.
We that's why we use some samples from our medical oncologist. We did, but we didn't put on the paper. But still, you can visualize them. But it is, but it is not like common that we use this. But you can do that, yes. But the thing is, it's gonna be showing kind of more blocks of regions. So our purpose was not like looking for them, but I think we should implement this like more efficiently, but we did that. And when we were testing, this was like not like we were testing for multi-patient VCF file. We were testing for single individual patient VCF files, and we visualized that. But I'm gonna put a note that maybe we need to make it more common use.
Yeah, that would be very useful. Also,
Yeah.
like comparing different samples, for example, like this amount of CNVs that they have and drawing
Exactly.
graphs.
Exactly. No, that's a very good suggestion. Thank you so much.
Great. Are there any other questions or comments from the participants here? All right, hearing none, I just want to say thank you again for coming and
Sure.
presenting this. And I definitely learned a lot, and I have some samples I want to try out with this tool. So thank you so much. Really appreciate all your effort and your time to come and teach us about this tool.
Thank you so much for having me. It was great to present here. Yeah.
Great.
Thank you.
All right. Have a great rest of your day.
Bye-bye.
Okay. Bye-bye.